let me share my screen with you all. So, alrighty. So, again, thank you so much for that generous introduction. I remember vividly, we met, I think, in Long Beach, California in 2009. And it was so nice to meet you. And I also want to thank everybody for joining in. I'm really excited to share with you all some of the work that my lab has been focusing on in the past few years. Right off the bat, if one looks at the transcriptome diversity across the tree of life, what becomes immediately clear is that while the number of protein coding genes that are shown here in white have remained relatively constant, the number of transcripts per gene, which are shown here in red, have steadily increased as the organisms evolved and became more and more complex. In fact, humans are now estimated to produce 7 to 8 transcripts per gene, many of which are expressed in a cell type specific manner and are regulated in response to changing physiological needs of a cell. So this is because in higher eukaryotes, one gene can give rise to multiple pre-messenger RNAs that have variable transcription start sites or polyadenylation sites. But the largest amount of transcriptome diversity perhaps arises due to the process of alternative splicing, where variable exons are spliced in or out in various combinations to increase the number of messenger RNA isoforms, and which can encode for a variety of polypeptides that can have different functions and sometimes even opposing ones. So the outcomes of alternative splicing are increase in proteome diversity, introduction of premature termination codons, which can cause mRNA downregulation by nonsense mediated decay, and also variability in messenger RNA 3' UTRs, which affects cis-acting elements that mediate regulation of messenger RNA translation efficiency, stability, and localization. And disruption of the activity of RNA binding proteins and also their cognate RNA targets has been now linked to many human diseases, including muscular dystrophies, heart disease, liver disease, as well as cancer. So one might ask, how does a cell know whether to include or skip an alternative exon in any given transcript? Now, it turns out, like everything else in molecular biology, there is a code for splicing. And the process of spliceosome assembly and the transesterification reactions must occur precisely on each intron of a primary transcript to form mature functional messenger RNA. Now, this involves the selection of authentic splice sites over cryptic splice sites and is determined by several factors that include splice site strength, the presence or absence of exonic and intronic enhancer sequences, exon-intron length, as well as RNA secondary structure. Now, splicing of constitutive exons is highly efficient because they're surrounded by strong splice sites. And these splice sites generally match the consensus sequence. The spliceosomal snRNAs can engage with these splice sites and then co-transcriptionally recruit the spliceosomal machinery to remove the intervening introns and ligate the neighboring exons. Now, alternative exons, however, have typically weak 5' and 3' splice sites, and therefore they rely on the presence or absence of additional cis-regulatory sequences within surrounding introns or sometimes in the exon itself. They also need what are called trans-regulatory factors, which bind to these cis elements and can thus either recruit or inhibit the spliceosomal assembly around alternative exons. Now, together, these cis and trans-regulatory factors constitute what is called the splicing code. Now, in my lab, we want to crack this code. First, we want to catalog all of the RNA variants that are coded in our genome. We want to determine their splicing patterns, their directionalities, temporal dynamics, as well as cell type specificity. Second, we want to uncover which regulatory factors, including RNA binding proteins, control these alternatively spliced events and how. And finally, we want to assess the biological significance of alternative splicing at a single gene and as well as the network levels. So while I don't have time today to tell you about our progress on all of these fronts, I plan to share a couple of stories to give you a glimpse of how developmentally regulated RNA splicing programs, when deployed properly, are highly effective in repairing and restoring an injured adult tissue. But on the flip side, in another context, this can be quite detrimental. Specifically, today I will describe the identification of a developmentally regulated alternative splicing program that rewires a critical signaling pathway to enable liver regeneration. Next, I will demonstrate that sustained reactivation of this developmental program drives pathogenesis of severe alcoholic hepatitis by overpopulating the liver with functionally immature cells. Now, this work was spearheaded by three very talented graduate students in the group, Sushant Bangru, Wakar Arif, and Joe Simets. All right, so Eduardo already sort of alluded to Prometheus's story. I will just say that one more time, that we all know mammalian livers have a remarkable capacity to regenerate, and perhaps that is why we can have a pretty good time at a bar on weekends and still show up to work and listen to a lecture on Monday morning. Turns out even the ancient Greeks knew about these awesome powers of the liver to regenerate. Stole fire from mankind, from Zeus and mankind. Zeus got really angry. He chained Prometheus to a rock. And as a punishment, each day an eagle would feast on a portion of Prometheus's liver. And each night his liver would grow right back for the eagle's return the very next day. So that's fine, but really, how does the liver achieve these amazing feats? So for a long time, regenerative capacity of the liver was attributed to a pool of facultative stem cells that can be quickly called upon to expand and differentiate into new hepatocytes or other parenchymal cells when needed. However, several lineage tracing and cell fate mapping studies in the past 5 years have shown that there are no stem cells in the liver. And in fact, following any sort of injury, I mean 99% of new hepatocytes are generated through self-duplication of pre-existing hepatocytes. Okay, so this is great, but still the underlying principles that trigger a quiescent hepatocyte to re-enter cell cycle and divide are still elusive. What is still unclear is that what molecular events drive reprogramming of quiescent hepatocytes into the proliferative state. How is the hepatic transcriptome, translatome, and proteome, if you will, remodeled to support proliferation while maintaining essential liver functions? And then what regulatory factors drive these remodeling events and how? So to answer these questions, we began by setting up a toxin injury and regeneration model in mice, where mice are fed the small molecule DDC in a diet, okay? And DDC is a hepatotoxin that inhibits the enzyme pherochelatase, which is a key enzyme in the heme biosynthetic pathway. Now, inhibition of pherochelatase results in iron overload, which causes hepatocyte death and compensatory regeneration response. Now, we first confirmed hepatocyte proliferation after DDC treatment by injecting a fluorescent EDU analog to label new DNA synthesis, which is a proxy for cell proliferation. And here in the immunofluorescence image of the mouse liver, the hepatocytes are labeled with HNF4 alpha in green, and the EDU positive cells are labeled in red. Now, while in the mice eating the normal chow, less than 0.1% of cells are proliferating. The number of proliferating hepatocytes in mice that have been fed DDC increased drastically demonstrating clear evidence of the compensatory regenerative response. Now, after we established the DDC-induced injury and regeneration model, we set out to systematically probe first the translation output of naive and regenerating hepatocytes. So we established a cell type specific in vivo polysome profiling method, and we combined it with high resolution transcriptome analysis to probe messenger RNA translation at a genome-wide scale. Now, I'm sure you all know messenger RNAs, when engaged with ribosomes, can be isolated as complexes using density gradient centrifugation, where messenger RNAs loaded with fewer ribosomes sediment in the lighter fractions, while the highly translating messenger RNAs are engaged with more ribosomes and therefore sediment in heavier fractions at the bottom. Now, before hepatocyte isolation, we perfused the livers with cyclohexamide to arrest the translating ribosomes, and then RNAs co-fractionating with monosomes, light polysomes, and heavy polysomes were purified and sequenced. We also developed a bioinformatics pipeline inspired by Eduardo for determining the relative ribosome occupancies of transcripts within a sample and the differential ribosomal occupancy of transcripts across experimental conditions, and which we termed as shifts in ribosomal occupancies, or Sherlock. And we're currently making some new updates to this pipeline, which we hope to publish and release in the near future. So when we compared the ribosome occupancies of messenger RNAs from naive, quiescent, and toxin-injured or regenerating hepatocytes, what we saw was there was some global reprogramming of translation that was going on in these regenerating hepatocytes. Interestingly, we found that liver injury reprograms hepatocyte transcriptome to an early postnatal-like state, okay? And translation regulation of alternative splicing factors activates a neonatal splicing program in these regenerating hepatocytes. Now, we also found that regeneration-regulated alternative exons frequently harbor amino acid residues that can be post-translationally modified to rewire protein-protein interactions. So next, we wanted to ask, how is developmentally regulated splicing reprogrammed in a regenerating liver? So from the list of alternative splicing factors that were translationally reprogrammed, that is, they were changing drastically in their protein expression, which you can see by these Western blots, but not really changing in their RNA expression, shown here on this graph. So we call them that they're all translationally regulated RNA binding proteins. We elected to focus on this protein called ESRP2. And this is because ESRP2 is a major determinant for neonatal to adult switch in alternative splicing in the liver. And also ESRP2 binding motifs were highly enriched around the regeneration regulated exons. So ESRP proteins were discovered as master epithelial splicing regulatory proteins in epithelial cells now more than a decade ago in the Karsten's laboratory. Now, there are two ESRP paralogs in mammals, ESRP1 and ESRP2, but only ESRP2 is expressed in the liver. And ESRPs bind to UG-rich RNA sequences near or within alternative exons to either promote or maintain their epithelial splicing patterns. So if you deplete ESRPs from epithelial cells, they spontaneously undergo epithelial to mesenchymal transition, and also the splicing pattern of many genes switches from epithelial to mesenchymal pattern. We have recently demonstrated that ESRP2 is a determinative factor for postnatal switch in alternative splicing of hundreds of genes within hepatocytes. ESRP2, as you can see here, is absent in the fetal and early postnatal stages of liver development. This is postnatal day 6, and this is postnatal day 28. But you can see that on day 28, it's already turned on, and actually it is turned on 2 weeks after birth. and is abundantly expressed in adult livers and adult hepatocytes. Now, the first 4 weeks after birth is a critical period of development when large numbers of developmentally regulated exons switch their splicing from fetal to adult patterns. Here I'm going to show you 2 examples of alternative exons that are developmentally regulated. In one case, this exon is absent in postnatal day 6 livers, but then it is included in the postnatal day 28 time point. And the other case, this exon is included more in the P6 stage, but then it starts to get more and more skipping at postnatal day 28. So now, if you knock out ESRP2 in the liver, these splicing transitions fail to occur, okay? And the P28 liver continues to express the fetal splice isoforms for both of these genes and many, many others, hundreds of other genes. So I asked my students, Sushant, that, you know, if ESRP2 is indeed determinative for these splicing transitions, you ought to be able to express ESRP2 ectopically in postnatal day 6 livers, normally when it is not expressed, and test if earlier than normal expression of ESRP2 is sufficient to turn on the adult splicing program in the P6 livers. So Sushant and my undergraduate student, Jackie, generated then a tetracycline-inducible ESRP2 bitransgenic mouse model, where we can drive the expression of ESRP2 using an RTTA that is driven by an ApoE RTTA, ApoE promoter, so which makes it sort of hepatocyte specific. And so we can, in this case, we can feed the nursing moms doxycycline containing food, and the bitransgenic pups receive that doxycycline through mom's milk, and this would turn on ESRP2 in these early stages of the liver development in these pups. So as you can see here, this strategy worked pretty well. Sushant and Jackie were able to turn on ESRP2 in P6 livers, which resulted in a strong switch in splicing of these genes to adult patterns earlier than normal. Now, these data demonstrate that ESRP2 is a major determinant for postnatal switch in alternative splicing within hepatocytes. In fact, my other student, Amruta, and undergrad Darren have determined that ESRP2 controls developmental switch of nearly 20% of all postnatal splicing transitions, and thereby it facilitates the terminal differentiation and maturation of hepatocytes. For instance, ESRP2 knockouts exhibit defects in cell cycle exit, and the knockout hepatocytes are considerably smaller in size relative to their littermate controls. Functionally, ESRP2 knockout livers are immature, and they produce significantly lower amounts of serum proteins and hepatic albumin. Now, let's revisit this earlier result where I showed you that ESRP2 levels are diminished following DDC administration. Now, to investigate if this downregulation following DDC injury is reversible, we set up an injury and recovery protocol in mice. So in this case, we feed mice DDC-containing diet for 4 weeks. And then after that, the cohort of mice are divided into 2 groups. In one group, the mice are sacrificed and the liver tissue is collected for histology and molecular analysis. Whereas the other group, we remove the DDC-containing diet and switch them back to normal chow for 4 weeks. And this is the recovery mice. So as you can see here, ESRP2 displayed a dynamic expression pattern during the injury and the recovery phase, where it's shut down after injury, but the levels are restored following recovery. But the key question for us was, does ESRP2 influence the proliferative potential of hepatocytes in response to injury? And as you might have guessed, the answer is yes. The livers of ESRP2 knockouts exhibit excessive hepatocyte proliferation upon injury. And when recovered, these livers or hepatocytes are unable to return to full quiescence because now ESRP2 levels cannot be restored. So you see high amount of proliferation in the injury phase, and in the recovery phase, while the wild type animals go back to their quiescent state, we have many cells that are still proliferating in the ESRP2 knockouts. Conversely, forced expression of ESRP2 during the injury phase blocks hepatocyte proliferation, resulting in impaired regeneration response. Now, among ESRP2 splicing targets were several genes involved in cell proliferation, including the core components of the hipposignaling pathway. Now, as you may all know, hipposignaling is an evolutionarily conserved pathway that governs the organ size and tissue regeneration in many metazoans. When hipposignaling is on, NF2 acts as a scaffold for the MSD complex, which phosphorylates YAP proteins to sequester them in the cytoplasm, and which also triggers their degradation by following phosphorylation by casein kinase 1D. Now, on the contrary, when hipposignaling is off, YAP1 accumulates in the nucleus and partners with the STEAD family of transcription factors to stimulate expression of many cell cycle and proliferation related genes. Now, strikingly, the core components of hipposignaling pathway, that is NF2, casein kinase, YAP1, and TEAD1, each contain a developmentally regulated alternative exon, which is predominantly skipped in the fetal or embryonic livers or hepatocytes, but these exons are included to a large extent in the adult hepatocytes. For instance, if you knock out ESRP2, these exons are skipped now, whereas if you overexpress ESRP2 in mouse livers, their inclusion is further increased. Now, liver injury by DDC administration mimics the ESRP2 knockout condition, okay, for each of these exons. And the exon inclusion for all 4 genes can be maintained if you sustain ESRP2 levels ectopically under DDC conditions. So this tells us that ESRP2 or these alternative exons are very sensitive to the ESRP2 levels in hepatocytes. Now, is this through direct engagement and binding of ESRP2 on these messenger RNAs encoding these hippopathway genes? So we wanted to do that, see that by doing the very common RNA binding protein RNA interaction assay called the CLIP assay. But due to lack of antibodies to immunoprecipitate ESRP2, we had to actually engineer a 2X flag tag at the N terminus of ESRP2 gene in mice using CRISPR-Cas9 genome editing. Now, this new mouse model provides us an excellent handle to pull down ESRP2 bound RNAs and also thus reveal ESRP2 binding sites across the transcriptome. Now, freshly isolated hepatocytes from adult flag tagged ESRP2 mouse livers were UV cross-linked, and lysates were prepared to immunoprecipitate ESRP2 using the anti-flag antibody. Now, as you can see here, I'm showing you 3 examples of the hippopathway genes, NF2, casein kinase, and YAP1. You can see that in the wild type mice, adult livers, these exons are included, right? And in the ESRP2 knockout, those exons are completely skipped, right? And you can also appreciate that flag ESRP2 eclipse shows that ESRP2 binds very close to these alternative exons, and this direct binding influences the inclusion of these alternative exons. So that's great, but the bigger question for us was, what are the functional differences between these neonatal and adult splice isoforms of the hipposignaling pathway? So we started to explore these isoforms further. Now, turns out that the neonatal isoform of NF2 can exist in two different conformations, a closed conformation and an open and active conformation. Now, LATS binding or the LATS binding site is accessible and hyposignaling is enabled in the open and the active state form, okay? However, if it is in its closed form, it cannot bind LATS2, right? So the C-terminal domain of NF2, when is interacting with the N-terminal firm domain, it sort of blocks the access of LATS and thus constrains its signaling activity. The adult NF2 isoform includes a 45 nucleotide alternative exon that I just showed you a few slides ago. And that 45 nucleotide exon is basically at the end of the C-terminal domain, okay, right here. And this turns out encodes for an early stop codon, resulting in truncation of 5 amino acids, 4 of which are known to be necessary for binding to the firm domain, okay? So the alternative exon inclusion now alters also the composition of 11 other amino acids from primarily hydrophobic to charged residues, okay? And now this is expected to create a repulsive force between the C-terminal domain and the hydrophobic binding pockets in the firm domain of NF2. So the adult NF2 isoform sort of now is in a state where it adopts a constitutively open state, providing uninterrupted access to LATS1 and 2, and thereby promoting YAP1 phosphorylation and degradation. So the end result in this case is that with the neonatal isoform, hyposignaling is going to be low, YAP activity will be high, and this is going to stimulate more proliferation. Now, however, with the adult isoform, hyposignaling is going to be high because it's in its open state, and YAP activity because of that is going to be low, and which will restrict proliferation, right? So next, if you look at the casein kinase 1D gene, the neonatal casein kinase 1D protein isoform encodes a C-terminal domain or a C-terminal tail that contains a stretch of serine residues, which when phosphorylated, lower its enzymatic activity. Now, elimination of these autoinhibitory phosphorylation sites enhances the enzyme activity towards these substrates. The adult casein kinase isoform, however, includes a 63 nucleotide alternative exon, okay, again in its C-terminal tail region. And just like NF2, this one also encodes for a early stop codon, truncating 6 amino acids. And notably, the alternative C-terminal tail now eliminates these 3 serine residues, okay? And in the adult casein kinase 1D isoform, this relieves the autoinhibition, thereby generating the more active kinase for YAP phosphorylation and degradation. Again, the end result is that the neonatal isoform with casein kinase 1D, the hyposignaling is going to be low, YAP activity is going to be high, and this is going to stimulate proliferation. However, with the adult isoform, now casein kinase is fully active, it is going to phosphorylate YAP. That's going to lead to lower activity of YAP and restrict proliferation in the adult liver. Now, let's go to YAP, the third component in this pathway. Now, YAP1 encodes a leucine zipper within its transcriptional activation domain, right? And this is important for mediating protein-protein interactions for YAP1. Now, the neonatal YAP1 isoform has the intact leucine zipper with 4 heptads, okay? However, the inclusion of a 48 nucleotide exon in the adult YAP isoform inserts this 16 amino acid stretch, which is intrinsically unstructured between heptads H3 and H4. This leads to its thermodynamic destabilization, and thus this leucine zipper is not favored. The formation of this leucine zipper is not favored. So the the inability of adult YAP1 isoform to form a functional leucine zipper is now expected to hinder its protein-protein interactions needed for association with other cellular partners. Again, in this case, the end result is that the neonatal isoform YAP activity is high, and which is going to stimulate proliferation. But in the adult isoform, YAP activity will be low, and this is going to restrict proliferation. Finally, the last component of this pathway is TEAD1, and turns out TEAD1 shares a conserved N-terminal T domain that binds DNA, okay, and a C-terminal transactivation domain. And this transactivation domain binds the cofactor YAP1, for instance, to induce target gene expression. Now, the T domain has 3 helix bundle, where H3 helix provides the interface for DNA binding. A 12 nucleotide microexon in the adult TEAD1 protein isoform adds these 4 amino acids just downstream of the DNA binding helix H3. And amongst these 4 amino acids is a conserved serine residue, which when phosphorylated, and this can be phosphorylated by protein kinase A or C, it strongly inhibits TEAD1's DNA binding activity. So you can see that the inclusion of this microexon reduces TEAD1's activity independent of its interactions with the cofactors such as YAP1, right? So again, at the end of the day, the result is going to be that with the neonatal isoform, TEAD1 activities will be high, and this will stimulate more proliferation, whereas with the adult isoform, TEAD1 activity will be low and proliferation will be low. So that's great, but we wanted to test this experimentally, whether this is happening. Now, because ESRP2 increases inclusion of all of these 4 alternative exons coordinately, we reasoned that ESRP2's influence on hepatocyte proliferation following injury might be driven in part by the production of the neonatal isoforms of hyposignaling pathway. So we decided to test this hypothesis directly. When hepatocytes are serum starved, okay, in culture, they drop their proliferation rates. And when released in minimal serum, they can re-enter the cell cycle and start dividing again. Now, we simultaneously redirected splicing of all 4 alternative exons in hepatocytes using antisense oligonucleotides that target either the 5' or the 3' splice sites surrounding these alternative exons. And this forced the production of neonatal isoforms from their endogenous transcripts. Now, relative to control ASOs, cells treated with a mixture of NF2, casein kinase 1B, YAP1, and TEAD1 targeted ASOs elicited a near complete switch to their exon skipped neonatal isoforms. And we next evaluated the functionality of splicing redirection on cellular proliferation. We released the starved cells in minimal serum following the treatment with control or antisense oligos that were targeting the hippo pathway. And we postulated that the cells expressing the neonatal isoforms of hippo pathway should display higher proliferation rates. And remarkably, exclusive production of the neonatal isoforms with these ASOs resulted in higher EDU incorporation as well as cell proliferation because we were able to show that these neonatal protein isoforms attenuate hyposignaling and also enable greater transcription of YAP-T target genes. Now, what I have shown you so far is that dynamic regulation of ESRP2 following liver injury reprograms alternative splicing to generate neonatal protein isoforms of hyposignaling pathway. And expression of the neonatal isoforms attenuates hyposignaling, activates gene expression, and promotes hepatocyte proliferation. So we can sort of revisit the old Greek tale now and say that after the eagle feasts on Prometheus's liver and leaves, the alternate hippos are redeployed to regenerate the liver. But as as it is true with most studies, what happened when we found this, that in order to regenerate, the livers had to turn on fetal splicing and fetal gene expression program. This led us to a new set of questions. We wanted to know, are all hepatocytes reprogrammed during regeneration to this postnatal fetal-like state? And more importantly, how do regenerating livers sustain normal metabolic activities as the tissue recovers from injury? Because you can imagine if there's already loss of hepatocytes in an injured liver, if you are going to turn on the fetal program, you're going to lose even more function, and that might be detrimental for the liver and for the organism. So how do these regenerating livers balance the metabolic activities while proliferation is going on? And we were also interested in knowing what cell-cell interactions initiate and terminate the regenerative response. So to answer these questions, we have recently leveraged a combination of single-cell RNA sequencing paradigm, and we've studied the initiation, progression, and termination phases of regeneration. Now, following a surgical resection or partial hepatectomy of the liver, the remnant liver tissue regenerates quickly and restores its original size and function within a week. Auinash and Sushant found that after doing the surgery, hepatocyte proliferation initiates in this mid-lobular region before proceeding towards the periportal and pericentral areas of the liver. They discovered that in order to balance the metabolic and proliferation needs, quiescent hepatocytes following surgery promptly adopt an intermediate state, okay? And they transition from this intermediate state into either the proliferative or the metabolically hyperactive states. Specifically, they found that a subset of residual hepatocytes in the mid-lobular area reversibly activate an early postnatal-like gene program that helps them to enter that proliferative state, while a distinct population of these portal and central vein, proximal regions, the hepatocytes there upregulate their metabolic gene program to offset any regeneration-induced deficits that occur when more cells in the mid-lobular region dedifferentiate and start to proliferate. Now, these reversible cell state transitions are guided by distinct ligand receptor-mediated signaling events between hepatocytes and non-parenchymal cells. And interestingly, the proliferating hepatocytes selectively downregulate the receptors for inbound signals. We believe that the elimination of receptors for inbound growth factors and pro-proliferative signals that arise from these non-parenchymal cells might be an important mechanism for limiting the endless proliferation of hepatocytes and enable the timely termination of liver regeneration. And we're very excited by these results and are following these up. But we were happy to see that along with our study, there were 2 additional studies published in Science in the same week, showing that it is the mid-lobular hepatocytes or zone 2 hepatocytes that are the major contributors to liver regeneration. And since then, many commentaries have highlighted these results, and there's a lot of excitement in the field to follow how these cells achieve how do they first re-enter cell cycle, and how do they terminate when the regeneration is complete. So in the last part, I want to cover an interesting idea that now if the liver's regeneration ability is so robust, why does it fail in a disease condition? So to explore this conundrum, we joined hands with Dr. Anna Mae Deal and her group at Duke University and decided to tackle this question in the context of alcoholic liver disease. Because excess alcohol consumption is a major cause of liver-related mortality in Western countries, severe alcoholic hepatitis, or SAH, is the deadliest form and includes a necro-inflammatory process with the fastest progression to cirrhosis. In fact, 1 month mortality rates in patients that are hospitalized with SAH is higher than many of the other fatal diseases, including heart disease, stroke, and most cancers. So normally, the adult factors, transcription factors such as CEBP alpha or cadherins such as CDH1 are highly expressed in normal, healthy livers, okay? Whereas the fetal factors such as YAP, TAZ, SOX9, and SMAD3, they are barely detectable in normal livers, right? In contrast, almost none of the hepatocytes in the livers of SAH patients express CEBP alpha or CDH1, okay? But on the other hand, YAP1, TAZ, SOX9, and SMAD3 are strongly upregulated. So we can appreciate that in severe alcoholic hepatitis, the livers are repopulated with cells that are enriched in progenitor and or fetal-like characteristics. In fact, when we sequence the RNA collected from the livers of SAH patients, the most strongly upregulated genes were related to functional categories enriched in inflammatory and oncofetal signaling, whereas the most downregulated genes were related to mature liver functions. So when we probed for ESRP2, we saw that expression of this adult splicing factor was significantly reduced at both the messenger RNA and protein levels in the livers of SAH patients vis-a-vis healthy individuals. And also ESRP2 was actually mislocalized. So you can see that in the normal, ESRP2 is largely nuclear, okay? But in the livers of severe alcoholic hepatitis individuals, the protein has moved from the nucleus to the cytoplasm. And as expected, the loss of ESRP2 was accompanied by accumulation of fetal kinase splice variants, which are relatively, I showed you, inactive when compared to the adult isoforms. So the big question was how and why is ESRP2 expression suppressed in SAH? So in the alcoholic liver injury field, people believe that excessive exposure to macrophage-derived pro-inflammatory cytokines such as TNF-alpha, IL-1 beta, and IL-6 drive SAH pathogenesis. But the question was now, does cytokine exposure affect ESRP2 expression? So we co-cultured adult mouse hepatocytes with mouse macrophages in a transwell setup and discovered that this both increases the hepatocyte exposure now to TNF-alpha that is being made by these macrophages. And it also suppresses ESRP2 expression in these hepatocytes. In a subsequent study, we treated hepatocytes, monocultures in this case, with TNF-alpha, IL-1 beta, and TNF-alpha plus IL-1 beta. And we found that these inflammatory cytokines directly suppress ESRP2 expression, indicating inflammatory cytokines are the likely cause for ESRP2 loss in the livers of severe alcoholic hepatitis individuals. So the bigger question now is, does ESRP2 suppression affects the pathogenesis of severe alcoholic hepatitis? So we set up to mimic the drinking behavior of the college students at UIUC. And in this case, what we decided was we took mice, and mice were initially fed a controlled liver decarly diet ad libitum for 5 days to acclimatize them to liquid diet, okay, and tube feeding. And afterward, ethanol-fed groups were allowed free access to the ethanol liver decarly diet that contained 5% volume by volume ethanol for 10 days. And the control groups were pair-fed with the isocaloric control diet. And at day 11, ethanol-fed and pair-fed mice were gavaged in the early morning with a single high dose of ethanol, 5 grams per kilogram. And or the control mice were gavaged with an isocaloric maltose dextrin. And 9 hours later, we euthanized these mice and then looked at their livers. Now, we examined if the hepatocytes of ESRP2 knockouts were more proliferative than the wild-type hepatocytes following alcohol-induced liver injury. So as you can see here, immunohistochemistry analysis showed rare cyclin D1 positive hepatocytes in the wild-type mice before alcohol feeding. However, these numbers increased in alcohol-fed mouse livers. Now, as expected, ESRP2 knockout hepatocytes were more proliferative than wild types at baseline, but the livers of ESRP2 knockouts became massively repopulated with proliferating hepatocytes after alcohol exposure. These hepatocytes also expressed fetal markers like SOX9. Altogether, these data suggest So these data suggest to us that ESRP2 depleted hepatocytes are primed to be reprogrammed into a proliferative fetal-like state. And this also confirms that loss of ESRP2 exacerbates fetal reprogramming that is induced by alcohol. So contrary to the conventional dogma, livers do not fail in patients with fatal severe alcoholic hepatitis because inflammatory cytokines are cytotoxic. Instead, what we believe is that these livers fail because inflammatory cytokines disrupt the molecular signaling programs that normally constrain the differentiation of hepatocytes. So normally, inflammatory cytokines are released by excessive alcohol ingestion, and they suppress ESRP2. And loss of ESRP2 now permits reemergence of fetal RNA splicing program that enables these hepatocytes to shed adult hepatocyte functions, become more regenerative, and heal alcohol-related liver damage. But now imagine if ESRP2 loss is sustained, such as the case in severe alcoholic hepatitis, because there is a continuous production of those cytokines that suppress ESRP2. Now, vital liver functions get compromised, and the liver becomes populated with functionally immature hepatocytes. So like I said, livers in the patients with SAH fail because inflammatory cytokines disrupt the splicing program that normally constrain the differentiation of adult hepatocytes. This causes functional parenchyma of the liver to be replaced by fetal-like cells, and these cells lack liver-specific functions. So with that, I would like to thank the people in my group who have done the hard work, and also our collaborators, Anna Mae Deal at Duke, Mikhail Hernandez at University of Navarro, Sai Anak, who's a liver expert at University of Illinois and gives lots of good advice whenever we need, and also the funding sources who have funded these projects over the years. I want to do a plug here for postdoc positions that are available in the lab. If any of you or your friends are interested in the sort of work that we're doing, please reach out to me, and we'll be happy to discuss some projects that we're very excited about. With that, I am happy to take any questions, and thank you so much for your attention.
Thank you very much for the great presentation. Is there any question from the audience? So then I will ask the first question. Okay. So you showed that co-culturing of liver cells and macrophage cells. So were those macrophage cells Casper cells? So they are normally liver resident macrophages? Or no, they were just in vitro differentiated macrophages?
They were in vitro differentiated macrophages, yeah. So we wanted to just do an in vitro experiment and see if cytokine exposure was sufficient coming from macrophages, and it was. So.
And how did you induce the pro-inflammatory phenotype?
Sorry, say that again.
So how did you induce the pro-inflammatory phenotype of macrophages?
No, so we were just culturing these macrophages we did not induce them. So macrophages naturally make TNF alpha, and that TNF alpha is enough to suppress ESRP2 in the hepatocyte cultures.
Sounds good. Thanks for the answer.
I have a question.
Yes.
Yep.
So I was interested in the role of casein kinase in regulating YAP. And I just wondered, is there any sort of genetic data coming along yet where they'll sort of knock out casein kinase, or maybe your splicing isoform? Could you genetically engineer a mouse with that sort of change and directly measure the YAP nuclear localization in the, say, liver with a liver-specific genetic knockout? Has anyone done that?
Thanks for that question, Barry. That's a great question. I don't think anybody has done that. In vitro, people have, sort of mutagenized, removed those, serine residues and showed that, but this experiment has not been done in a mouse. But that's a great idea. I mean, I think it will probably tell us how important those residues are in controlling phosphorylation of YAP, or whether there are other kinases that sort of compensate now. Sure. So as we know, there are many kinases that phosphorylate YAP. So, but we were very happy to see that the whole pathway is, there are these alternative segments that are being put into the whole pathway to sort of shut down proliferation, and it's a very coordinated sort of regulation. When we found that, we were pretty excited. So we have made with CRISPR some alternative exon knockouts, but we have not done that with the hippo pathway yet. But that's the plan, is to at least start with some. Casein kinase may not be your first one that we go for.
Sure. But
we're thinking of using the NF2 as first one. But yeah, that'd be super. Thank you.
Great.
I have a quick question. It's probably a silly question, so I'm not an expert, so but I'm wondering. So is this also like a model to study what the for the next steps would be to develop liver cancer, or what are the additional factors that may play a role in that?
That's a great question. So a lot of times these people who have been drinking alcohol for decades, they end up with liver cancer, and there are also other diseases where there is excessive proliferation, excessive regeneration that occurs, and that also unfortunately ends up with liver cancer. So we have actually done some experiments with the ESRP2 knockouts. Although if you just normally age the ESRP2 knockouts, they don't get cancer spontaneously. But if you give them any sort of a agent that causes DNA lesions, such as DEN is a well-known model. You can inject DEN, which causes mutations in the DNA early on, and then there is normally the wild-type mice will develop tumors and full-fledged cancer by a year or so. And if you do the same experiment in the ESRP2 knockouts, they develop cancer by 8 to 9 months. So it accelerates the development of tumors and cancers. It's not sufficient to cause cancer, but if somebody had low ESRP2 activity, they're more primed. Because they're more proliferating, they're more primed to get cancer. All they need is some mutation, and the cells could just take off.
Okay, that's really exciting. Thanks.
Thank you.
Thank you very much. I have another question as well. So do you think that's a general phenomenon throughout the body related to diseases, that the resident macrophages secrete pro-inflammatory cytokines, and that causes the differentiation of the tissues?
Yes. Thank you for that question. I believe so. I think a lot of times we know that inflammation is a key mechanism for tissues to recover, right? Whether that is from infection or injury. And people have focused on largely the cytotoxicity of inflammatory cytokines, that they are going to kill the the injured cell, or they're going to kill the invading pathogen or whatnot. But I think another important aspect of these cytokines is also that they reprogram the cells around where they are produced so that they can help with the healing, help with the recovery. So, and I feel that this is going to be a common theme in most tissues where there is some sort of repair mechanism that is available. I'm not sure whether that happens in the heart or whether that happens in tissues like brain, but cells that have the capacity to renew, self-renew, they might be waiting on these sorts of signals. And of course, if there is a chronic inflammatory sort of a situation, this can turn on its head, and now you have these signals being made all the time, and then they could lead to either over proliferation, over stimulation, and also, if you will, over fertilization of the tissue, and that could be bad for the tissue. Yeah. So there has to be a very nice balance between how much and when to stop.
Thank you very much for a great answer. Is there any other question from the audience? So if we have no more questions, thank you very much for the great presentation.
Thank you. Thank you.
We were happy to have you here, and hopefully we can catch up in person very soon.
Absolutely. That would be fantastic. And good luck with your research and your projects. And I'm looking forward to meeting with you guys. And of course, Eduardo, next time, whenever, man.
Yeah, let's hope it's soon. Yes, that'll be great.
Yes. Thank you so much for hosting me. Yes.
No, thanks a lot. It's been great. Thanks a lot for the great talk, and I hope to see you soon. Yeah,
okay.
And
take care.
yeah, take care.
Bye bye.
Stay safe. Bye bye.